Examiners are consistently reporting that Cambridge Biology 9700 Topic 1.1 Resolution versus Magnification in Light and Electron Microscopy is literally one of the single most misunderstood areas of the entire syllabus. Well, we're going to fix that right now, giving you exactly what senior examiners are actually looking for.
So, ask yourself this. What is the one critical error, the single biggest mistake that is silently stripping top marks away from hardworking students in this exact topic? It's the kind of error that creates an absolute instant confidence shock when those results come out. You know what it is?
Generic answers. Okay, let's dive into this. Because the ultimate grade killer here isn't a lack of revision. It's failing to apply your knowledge of microscopy concepts to the unfamiliar, highly specific contexts they throw at you in the exam. Honestly, memorizing the textbook is completely useless if you don't know how to adapt that knowledge on the fly. So here is the procedural framework we'll use today to turn that around. 1. The costly exam mistake. 2. Mastering core principles. 3. The method mark strategy. 4. Acing comparative analysis. And 5. Top examiner warnings.
Let's start with Section 1, the costly exam mistake, losing marks to vague context.
Listen to this, straight from the principal examiner reports. They state verbatim, the classic mistake is providing generic answers not specific to the question context. Always link every point to the given scenario. So knowing the syllabus parameters for an electron microscope, that is nowhere near enough. You have to know how the examiner thinks and exactly what the stimulus material is demanding from you. And this brilliantly illustrates the difference. If you look at the flawed student approach, it just spits out general facts, right? Just stating that electron microscopes have a higher resolution than light microscopes. I mean, it's true, but it's totally generic. Now look at the top band examiner answer. It connects the specific principle of resolving power directly to the visualization of the precise organelle mentioned in the stimulus scenario, like the inner membrane of a mitochondrion. That is how you score the marks. Moving right along to Section 2, Mastering Core Principles, Theoretical Architecture. This theoretical architecture rests on 3 main pillars. First, precise definitions. You absolutely cannot blur the lines between resolution and magnification. Second, causal relationships. You need to understand exactly why a shorter wavelength results in higher resolving power. And third, and honestly this is the most important one, integration with other syllabus areas. The data actually shows that candidates who reference cross-topic connections, like linking microscopy limitations to cellular transport mechanisms or cell division, they achieve significantly higher marks. All right, Section 3, the method mark strategy, procedural framework. Now this is your ultimate quick win in this explainer. What a lot of really focused learners don't realize is that examiners actually award marks for the analytical process itself, even if your final conclusion is slightly off. So treat these 5 steps like an absolute checklist every single time you see a microscopy data question. Step 1, deconstruct the command words in the question. Step 2, extract the data directly from the stimulus. Step 3, select the correct analytical framework. Step 4, execute your calculations with crystal clear notation. And Step 5, present your findings with perfect terminology. If you do this, you capture every possible method mark available. But pause for a second. You might have the steps down perfectly, but what's the hidden examiner clue that changes your analytical framework completely? In just a moment, I am going to reveal the exact phrase that dictates how you must analyze the data. But to even spot that hidden clue, you've got to completely understand your analytical tools first. Now, what's really interesting about this slide is how it cuts right through the cognitive overload of statistical analysis. When evaluating microscopy data, you must choose the right tool for the job. If the clue in the stimulus asks you to look at averages between 2 different cell samples, boom, use the t-test for comparing means. If you're looking at frequencies of observed organelles, it's chi-squared for categorical data. And if you are assessing how changing magnification impacts your observed field of view, you're looking at correlation tests for relationships between variables. It's really that straightforward once you map it out.
Let's jump into Section 4, Acing Comparative Analysis, Critical Evaluation.
This brings us to the real climax of our explainer, where we deal with the highest order assessment objectives that demand critical evaluation and justified conclusions. Let's see how this builds up our analytical toolkit. When you're comparing light and electron microscopes, you must identify similarities and differences. So many candidates list the similarities but completely forget to evaluate the differences, or vice versa. You must use comparative connectives like whereas or in contrast. You've got to support each individual comparison with evidence from the provided text or image. And finally, draw conclusions about their relative importance to that specific biological context. Look at this comparison right here. A descriptive level answer simply lists basic facts about the microscopes, but an evaluative level answer, which is the elite approach taken by top-scoring candidates, actually questions assumptions, considers different perspectives based on the specific specimen being viewed, and justifies exactly why one microscopic technique is absolutely superior to the other for that specific investigation. And they back the whole thing up with evidence.
And finally, Section 5, Top Examiner Warnings, the final checklist.
So we've looked at the strategy, but now let's look at the traps. There are exactly 3 major mistakes repeatedly flagged in the principal examiner reports year after year for Topic 1.1. If you can eliminate these 3, your grade boundary risk drops significantly. When we look really closely at those examiner reports, these 3 main traps pop up constantly. Trap number 1, confusing cause and effect. Please do not mix up the reason for high resolution with the outcome of high magnification. Trap number 2, forgetting to check your units and terminology. Converting micrometers to nanometers incorrectly is just a devastating, unforced error. And trap number 3, as we hammered home earlier, your answers simply aren't specific to the context given.
Seriously, I cannot stress this enough. Check your units. It literally takes 2 seconds of cognitive focus, but missing a simple conversion from millimeters to micrometers will instantly void your final calculation marks. Don't let that happen.
Add link to the scenario. Do not give the examiner a textbook definition if the question is specifically asking about a palisade mesophyll cell. Talk about the palisade mesophyll cell. So the crucial point is this. You need a reliable way to lock this all in under exam pressure. Here is a really practical memory hack. Create a mnemonic using the first letters of your key microscopy terms: resolution, magnification, wavelength, electrons, and photons. Build a weird or funny phrase that sticks in your mind so that when the clock is ticking and the pressure spikes, your recall is automatic and perfectly structured. Okay, you now have the framework to avoid contextual mistakes, utilize those method marks, and execute top-tier comparisons. But there is one final piece to this whole puzzle.
What are the exact command words that actually decide your final mark band? That is the hidden diagnostic trigger we'll be solving next time. Subscribe to our YouTube channel, Ace the Exams, at acetheexams_IGCSE to excel in your Cambridge examinations and find out exactly what they are. See you there.